Hello, everyone, and welcome to today's live broadcast, Immunotherapy, a Revolution in Cancer Treatment, presented by Brad Nelson, Ph.D., Distinguished Scientist and Director, Deeley Research Center, Co-Director, Cancer Immunotherapy Program, BC Cancer Agency. I am Christina Mahalik of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone that this is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into that box that appears on your screen. We will answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Now, please join me in welcoming Dr. Nelson. I will now turn the presentation over to him. Dr. Nelson?
Well, hello, everyone, and thanks very much for the introduction, Christine. We're here today to talk about immunotherapy, which genuinely is a revolution in cancer treatment. If you haven't heard already, it's really changing the way cancer is treated in the here and now, and it's going to have a major impact on how cancer is treated in the future. So just to add a little bit more to my introduction, I work at the BC Cancer Agency that's based in British Columbia, Canada. Our program, I co-lead it with Rob Holt, who's at our Genome Sciences Center, and we're divided between two cities, Victoria and Vancouver. And I'll talk a little bit about what we're doing, although I'm going to keep this at a pretty high level, really talking more about what's going on around the world. Immunotherapy, in a nutshell, is illustrated in this cartoon. What we're trying to do is get immune cells, in particular T cells, to attack tumor cells. And so this cartoon illustrates it very simply. We've got a T cell on the left there, that purple-ish cell. I'll tell you a lot more about these, but basically these are a type of white blood cell that we all have in our bloodstream. Protects us from infections all day, every day. But T cells also have the ability to recognize tumor cells. And just as shown here, they can literally attach themselves to the tumor cell. They can poke holes in it and inject toxic enzymes and other compounds to just cause the destruction of that tumor cell within minutes of contact. So with immunotherapy, this is how we're trying to fight cancer. There are all sorts of different, what we call modalities or approaches to immunotherapy. This wheel I borrowed from a review article that was published a few years ago, just really illustrating the different approaches involving antibodies, vaccines, viruses, T-cell infusions, etc. And through the course of this presentation, I'll touch on a few of these different modalities. But just wanted to start by giving you the big picture that immunotherapy is not one approach, but it's a whole suite of approaches, all of which have the same goal, which is to enhance the immune response to cancer. So the topics I will cover today are, first, we'll just step back a bit and talk about how does the immune system respond to cancer in the first place. Just what is the natural response? What does that look like before we even try to intervene? Second, I'll talk about immunotherapy and really highlight why it is now the hottest field in oncology. and it's not just me saying that, by the way. It's really generally widely accepted now, and you'll see the breakthroughs that have been made. This really is where the big advances are happening right now, and I'll tell you why. And then, of course, what does the future hold? We've had some success now, but what do we see looking forward from here? What successes and challenges lie ahead? So starting at really the beginning of our discussion here, let's just think about how cancer develops, and that will help really frame the discussion about the immune response and immunotherapy. So starting on the left, we have just some normal tissue. This could be your skin, it could be the lining of your lung, your gut, but you've got a sort of normal cells, and in the second panel over, we see something's happened. There's been some genetic damage, perhaps environmental, perhaps other reasons, but a cell indicated in blue there has become, its DNA has become damaged and mutated or changed in some way. And that can then lead to what we call dysplasia, which is these benign lumps. We all have a number of lumps and bumps in our body that are benign, dysplastic tissue, not too worrisome on their own. But those, if unchecked, some of those can then progress to cancer. And the first stage of cancer is what we would call an in situ carcinoma. And this is, again, earliest stage of cancer. We use the word in situ, the term, because it indicates the cancer has not spread anywhere yet. It's still in the same situation or location as it began. When cancer becomes challenging is when it becomes invasive and starts to break out of its site of origin and eventually starts spreading to neighboring tissues, and if unchecked, it can then start spreading around the body, and which we call metastasis, and that's when it's very challenging to treat. So with in situ carcinomas, let's just step back to that stage. Those are highly treatable with today's treatments in general. So a surgeon can cut those lumps out. You might give a little bit of local radiation to clean up any residual cells, but generally those are easily treated. It's when a cancer becomes invasive and metastatic that it's very difficult to treat, because, of course, the surgeon can cut out the tumors that they see, but they're unable to remove every tumor from the body. And so we have to come up with what we call systemic therapies, treatments that are body-wide, treat the whole from head to toe, on the assumption that cancer cells can be hiding anywhere. And that's when we start using agents such as chemotherapy to treat the whole body. And that's where immunotherapy, as we'll see, plays a big role, because the immune system is everywhere and has the potential to control cancer wherever it might be hiding. So just at a high level or simple level, what's happening as cancer develops is, in general, that there's an accumulation of mutations in these tumors as they progress. And those mutations appear to occur more or less randomly. And then there's a process of Darwinian selection, much the way species evolve on our planet. Tumors evolve through these random mutations, but then tumor cells are selected upon. Many tumor cells die off. They get mutations that are actually harmful to them. But once in a blue moon, you get just the right constellation of mutations that, rather than causing the tumor cell to die, they actually give it these superpowers to start growing out of control and forming these invasive cancers. So there's a real random element to all of this, which again makes cancer challenging, because no two cancers are exactly alike. The next slide shows you what I mean by mutations and just how corrupted or disrupted the cancer genome can become. So first of all, the term genome refers to the set of chromosomes or genes in a cell. On the left, we're looking at a normal cell. It has the normal complement of 23 chromosomes. They're nicely paired up there. You can see 2 copies of chromosome 1, of chromosome 2, etc. 1 copy comes from your mom, 1 from your dad at the time of fertilization. So that's what the healthy cell looks like. And on the right, I'm showing the chromosomes from a typical cancer cell. What you see is we've got something like 5 copies of chromosome 1 shown there, 4 copies of chromosome 2, etc. So you actually can have gains in the number of chromosomes. You can also have losses of chromosomes or sections of chromosomes. You can also see there are some, let me get my pointer going here. Here we see examples of a multicolored chromosome. That's a case where the chromosomes have been cut apart and pasted back together in abnormal configurations. So if you imagine that this was your laptop, the software for your laptop undergoing these kind of changes, you might think your laptop would crash as a result. And again, probably these kind of mutations do lead to cells crashing and dying most of the time. But once in a blue moon, you get just the right combination of mutations that actually leads to a highly viable cell which starts to cause the problem of cancer. So again, because it's a random process underlying cancer, the result is that no 2 cancers are alike. So I'm showing you now chromosomal pictures from 6 different cancers. They're all breast cancer. And you can see just at a high level that they all have in common the fact that they are very messed up, lots of changes, but the details of those changes, those aberrations, are different from case to case. So when a patient walks in the clinic with cancer, at some level one could say their cancer has never been seen before. It'll be a unique entity. Now, it's not quite as bad as that might sound in that, especially with modern genomic technologies, we can go much deeper than this and look at mutations right down to the level of individual letters in the genome, and then we can do sophisticated computational analyses of these bioinformatics and find that there are patterns to all this. The successful cancer cells share common patterns of mutations, different pathways that are being activated, and so that provides hope that there is some sense to all this, and that therapeutic approaches can be developed in a rational way. But nonetheless, there still is this random element of the background that presents a challenge to any type of cancer treatment, including immunotherapy. Adding further to the complexity, cancers are not just, as indicated here, a homogenous entity. It's not just a ball of monoclonal homogenous tumor cells, but rather, because of the way cancers evolve, it's much like species on the planet. You end up with families of cancer cells in a given patient. They're all related. They all came from the same grandfather or great-grandfather cell way back in time, but they've acquired additional changes that make them unique. So on the, I'm showing an example from a patient with ovarian cancer. This is from a study of a colleague of mine that was published last year, but showing here the different tumor clones, as we call them, that are found within this patient. So the patient's cancer started with something that looked like clone A, which then over time gave rise to a number of other clones which are related but have unique mutations within them as well. And on the right, from the study, what they did was looked in the peritoneal cavity of this patient where ovarian cancer resides, and they sampled tumors from different regions, from the left ovary, the right ovary, pelvic regions, metastatic sites, etc. And they, using genomic and bioinformatic tools, were able to tell which of these clones from the family were found at different sites. And so you can see, for example, in this left pelvic region, the tumor there was composed primarily of clone B from this family, whereas if we look down in the right ovary, the cancer that was found there was composed of clones G, some E, and a little bit of H thrown in there. So again, these clones are all related, but have unique mutations and unique properties. So when we think about trying to treat this patient, again, this is all data from one patient, we're really having to come up with therapies that will treat and kill all of these clones, not just some of them. It would, in theory, just take one clone to survive, and the patient would still be struggling with their cancer. So this is the enormous challenge that we face when trying to treat cancer. Just kind of recapping that, I think a useful analogy might be to think of using the mafia as an example here. You don't just have one bad guy in a cancer patient, but you've got a whole family, and they're related, and they probably even work together to carry out their bad deeds. But there are some good guys in this story, as indicated by Charlie's Angels here. And this is, of course, I'm going to talk about the immune system and how it tries to fight back against this corruption. So the immune system is basically all your white blood cells. We all have them. They, as I said earlier, they help fight infection all day. But it turns out the immune system can also recognize cancer. So I'm showing you now an image of ovarian cancer. Again, this has generated this picture in my lab from one of our patient samples. And what we see is in purple, first of all, those are all the tumor cells, all the bad guys that are forming the tumor in this patient. And in brown, we see these so-called killer T cells. These are the cells I talked about at the very beginning that are able to go and latch onto a tumor cell, poke holes in it, inject toxic compounds, and kill it. That's why they're called killer T cells. We also have some helper cells in there in green, and some B cells in pink. B cells make antibodies, and we think those also help to fight the cancer. So you can see in this actual image, not live, but real image from a real patient, you can see her immune system is actually infiltrating the tumor, much like foot soldiers going into a battlefield and engaging in hand-to-hand combat with these tumor cells and fighting away. So in most patients, we see this kind of response to some extent. I'll say a bit more about that on the next slide. Actually, I'll just go to that. I'm showing you now similar types of images, but we're looking at 3 different patients now, just to show you the diversity of responses we see when we look at multiple patients. So 3 different tumor samples being analyzed here, and you can see on the left the color code. So here we're staining for, again, the killer T cells are in sort of a brownish color, the helper cells are purpley in this case, and the B cells are once again pink. So you can see, let's start on the far right here, that this patient has a very strong immune response, we would say, as indicated by this battle that's going on. In the middle, we have a patient with a moderate immune response, fewer T cells, and they tend to be stuck in the stromal regions of the tumor rather than the actual malignant part of the tumor. And then this patient on the far left has a very weak immune response. In fact, her tumor looks, in this case, negative for any sort of immune response. So I'll just make it clear, we haven't done anything to cause these patterns. The patients have just walked in off the street for their first surgery. They've just been diagnosed with cancer. And so this is a snapshot of what their immune system is doing naturally to fight their cancer. This is the starting point. And we're, of course, very interested in this. Why do some patients have a strong immune response, like the one on the right, and other patients, the immune response is much weaker? This is important for a number of reasons, but the main one is shown toward the bottom of the figure here, which is if we look at survival rates of patients and we just score them according to the strength of their immune response, you can see that with these moderate immune responses, on average, patients have about a twofold better chance of surviving. And with these strong immune responses, that's about a threefold better chance. This is, I should clarify, I'm giving you information just for ovarian cancer. Those are what the numbers look like. But the same pattern, the same thing applies to virtually every other type of solid cancer, where the presence of a strong immune response is a good thing. It leads to survival. So we've known this for about 10 or 20 years now, and this has really provided inspiration for thinking about, okay, well, what can we do to enhance these immune responses? How do we make a strong immune response even stronger? And how do we get an immune response started in somebody where it's a weak immune response to begin with? So with immunotherapy, that's all we're trying to do, is really understand these responses and learn ways to amplify them so that those survival rates can go up even higher. So what's holding back the immune response? You know, if you've got a strong immune response, why do you have cancer at all? Well, it turns out, like any biological system, there are negative feedback loops in the system. And so the immune response is no different. There are both stimulatory and inhibitory signals that these immune cells are being guided by. And a really important set of signals is shown in this illustration here. So I'm showing a tumor cell on the upper cell and then a T cell in the lower cell. And get my pointer back here. One important signal is the antigen triggering activation of the T cell receptor. That turns the T cell on and gets it activated. But that signal is opposed by negative signals in a particularly important pathway. Not the only one, but a very important one is called the PD-1, PD-L1 pathway, which actually sends a negative signal to the cells. So to illustrate this, we can think of the gas pedal and the brakes on your car. The antigen and T cell receptor interaction is like putting your foot on the gas. That activates the T cell. The PD-1, PD-L1 interaction is the brake. It's like putting your foot on the brake and basically slows down or impedes the T cell response. And this is what this brake looks like in the real world. So back to ovarian cancer, which I'm using as our example today. We're looking at a tumor now, and this time we've stained it for two different things. We've got our T cells, the good guys are in green, so you can see those green cells. By the way, these colors keep changing just because we use different staining methods to reveal these cells. The cells themselves are white blood cells, as I said earlier. But here they're looking green, the way we've stained them. And then in brown, we're staining for this PD-L1 molecule, which is part of the braking mechanism I just mentioned. And we see these PD-L1 positive cells, which we know by other means are a cell type called macrophages. Macrophages are normally good guys. They are kind of like the garbage men that go pick up the garbage throughout the body, dead cell debris, etc. But in this case, the macrophages, due to the biology here, are starting to express this PD-L1, which is providing a brake that's actually slowing down or stalling out those good guys, the CD8 positive T cells. And you can see that almost everywhere that we see a T cell in green, it's surrounded by a cloud of these PD-L1 positive macrophages, obviously making it very difficult for those T cells to do their job and actually attack the tumor. So it's a bit like being in a car with the driver from hell, if you will, who has their foot on the gas and on the brake at the same time, and you're sort of just lurching forward in traffic, but really not getting anywhere productively. So you could think of that as a stalemate. So with immunotherapy then, we seek to enhance these responses. We want to take the foot off the brake and put more on the gas. We want to break through stalemates, etc. And this is the figure I showed you before, just as a reminder that there's lots of different ways to think about doing that. And I'll start working through a few of those examples now and show you some of the successes. So the first is just really building on what I've said just a minute ago with this gas and brake analogy, is to take the foot off the brake. And the way that's done pharmacologically these days is various groups have come up with antibodies, illustrated here, that an antibody to PD-1, for example, or in yellow, an antibody to PD-L1, which can come in there and basically block that interaction between PD-1 and PD-L1 and disengage that brake. So you now have a scenario where, in this case, you've got your foot on the gas still. There's our gas pedal, but you've disengaged the brake. And so the T cell now is activated. It's free to go and attack that tumor cell and kill it. I'll just point out that in practice, you don't need both of these antibodies. You wouldn't need to block both PD-1 and PD-L1 at the same time, but rather these are two different drugs that hit the same pathway. So this approach is called checkpoint blockade for various reasons. We think of these breaks, others refer to them as immunological checkpoints. So with checkpoint blockade, we're blocking those checkpoints. And here are some of the clinical successes that have been reported around the world just in the last 5 years or so. I'm not here to promote any particular drug name, but these are just some of the common drugs that are out there that patients are hearing about and knowing about. So I've thrown some of those names up there, but in no way intending to promote one over the other. But I talked about the PD-1 brake. One that I didn't mention, but you can see at the top there is another braking mechanism involves a molecule called CTLA4. And that was actually, that drugs against that target were first out of the gate and received FDA approval for melanoma a few years ago now. As far as blocking the PD-1 pathway, there's a number of drugs out there that can do that. And you can just look down this list and see the wide variety of cancers that are responding to this type of treatment. And I've highlighted in blue the FDA approvals that we've seen just really in the last few years. So this is why we're talking about this being a revolution in cancer treatment. This is a lot of FDA approvals across a wide variety of cancers in a really short period of time. And it's really just become a game changer for oncology. A couple of things I'll highlight, for example, with Hodgkin's lymphoma, with this approach, there's about an 87% objective response rate, which is just absolutely remarkable. Recently had an FDA approval for one of these agents, one of these drugs for treating any adult or pediatric metastatic solid tumor that has a particular type of DNA repair issue called mismatch repair deficiency. So this is a very sweeping approval that cuts across all types of cancer that have this genetic aberration in them. And that's really an unprecedented type of FDA approval. And then at the bottom here, I'll just point out that these agents are now starting to replace frontline chemotherapy for melanoma and lung cancer, and in all likelihood other cancers, and many other cancers will follow suit. So this is the revolution that I'm talking about, where we're actually seeing chemotherapy being replaced now by immunotherapy. And for those of us that have worked in the immunotherapy field for several decades, such as myself, I think we are asking ourselves, did we ever really expect to see this in our lifetime? I mean, this is just really fantastic progress that we're very excited and encouraged by. So, of course, everything brings challenges. So what are some of the challenges with checkpoint blockade? Cost. These new drugs are coming in at about $100,000 per treatment cycle, just using rough numbers, but they're considered expensive drugs. For some cancers, combinations of these drugs may be required. So two drugs, Ipi and Nivo, are now recommended for use together to treat melanoma in some cases. So that doubles the cost as well as any side effects that may come along. And then for these drugs, the big question is, how long do you need to keep a patient on them? If they're responding, do they need to stay on the drug, or can you stop giving the drug? You might think, well, if you activated the immune system, maybe it can do the rest of the job on its own and the drugs are no longer required. Or you might think, well, no, if you are getting your foot off the brake, you got to keep it off the brake for as long as possible. So this is a big issue, is how long do the patient need to stay on these agents? And length of time, of course, adds to cost and side effects. The other big issue is under efficacy, or how well they work. So many cancers, I've given you some of the best examples, but there's other cancers such as ovarian and breast where the response rates are still disappointingly low, down in the 10% range or so. And also the responses are often transient. And with lung cancer, there's a lot of success here, but for most patients, it doesn't appear yet that they're being cured by these drugs. They're getting a lot of benefit, but it may not be forever. So there's a lot of work still to be done then on these issues. And this slide shows just really how complex the interactions are between the immune system and cancer, but also all the other opportunities that lie ahead for us. So this is looking at all the gas pedals and brakes, or most of them, that we know about, that make up the dialogue between T cells and their target cells. So I've talked about PD-1, PD-L1 pathway, and there it is up near the top of the list. I've also mentioned the CTLA-4 pathway. There it is. But you can see there are a lot of other molecules involved in this dialogue. And in this context, it's really actually quite remarkable that just disrupting one of these braking mechanisms has any influence. I've just shown you how effective these two pathways can be when targeted. Quite remarkable when you put it in the context of all the other signaling pathways that need to be considered. So the field right now is very excited and working through some of these other gas pedals and brakes and learning, developing drugs against them and learning how to use these in combination and really learn how to drive these T cells as effectively as possible. So T cells, an analogy I use, they're not like an old biplane, not like the first airplanes that first came out that just had very simple steering wheels and et cetera, but they're more like a 747 with a very complex dashboard And that's good because that allows our immune system to do very sophisticated things. But it also means that some work is going to be required to really learn how to drive them. So this is another view of this, what we call the synapse, or the space, the communications channel between here. Now the T cell is on the bottom and the target cell is on the top, and just showing some of these molecules again, and then in purple indicating where some of these checkpoint blockade antibodies operate. So that's what we've already talked about. But I'm showing this now to just kind of point out how these are working inside the T cell. So all of these molecules down below are the intracellular circuitry that T cells use to interpret the signals coming through these gas pedals and brakes. So these are, part of the brains of the T cell, if you will, that take all of this information, collect it all, and then ultimately make decisions for the T cell about, okay, are we going to attack this tumor cell or not? So all that information gets integrated into some very complex intracellular signaling machinery. And there's opportunities there as well then for therapies where one can think about coming in with drugs that modulate these signaling pathways is another approach to changing the behavior of the T cells. And I'll show you an example of that later. But how do we do this in a really rational way? And when we think about the future, is the future going to be giving patients all sorts of different antibodies, as we're currently doing, that affect these signaling pathways? Or can we be thinking about genetically engineering the T cells, changing these intracellular signaling circuits so that the T cell actually starts behaving differently? And I'm a proponent of the latter approach. I think the future of immunotherapy for cancer is going to involve actually getting in there and genetically engineering the T cells to change their response characteristics. And I'll say more about that and also make the case that it's doable, it's yielding some early successes, and there's huge opportunities. So back to our wheel here, what I'm going to talk about now is something called adoptive cell transfer up in that quadrant. And this is where the opportunities lie for doing these types of genetic engineering approaches. So I'll introduce the topic of adoptive T cell therapy, often called ACT for short. And this is really a variety of approaches, but they all follow the same basic concept here, but there's lots of flavors. But basically it starts by collecting T cells from a patient from either their tumor or a blood sample. You can then identify, or as we'll talk about, genetically engineer cells to recognize the tumor. You then expand the T cells, typically in the lab, to reasonable or large numbers. And then those T cells can be infused back into the patient's bloodstream just through a simple IV bag. And often we would give drugs along with them to help them out, such as the checkpoint drugs we've talked about. But those T cells, once they're back in the bloodstream, can circulate throughout the patient's entire body, and they can be then able to recognize and respond to destroy the cancer cells wherever they might lie. So this is a great opportunity to really take control of the immune system, change its properties, and create just a supercharged T cell response. And just to show you how powerful this can be, this is data that my group published a number of years ago now, just in a mouse model of breast cancer, where we did T cell therapy just with some pretty simple maneuver. We just basically increased the number of tumor-reactive T cells in the mouse. We didn't genetically engineer them or anything fancy like that. But just by increasing the number of T cells to supercharged levels, you can see the tumor on the left is an untreated tumor. This would be like some of the human tumors I showed you, where there's really not much of the immune response going on. And then panel B shows what a similar tumor would look like 7 days after giving adoptive T cell therapy. And all of those little brown cells you see in there, hundreds or thousands of them, each one of those is a T cell. You can see that has just very strongly, profoundly invaded the tumor tissue. And in fact, what we see in these mice models is that the tumors actually swell up for a few days after T cell therapy because they become so chock full of T cells really overtake it, and then the tumor collapses over the next few days. And by about 10 to 14 days later, in these mouse models, the tumors actually completely disappear, just eradicated by the T cells alone, and never come back. Now that's in mouse models. I'm not claiming it's as simple as that in people yet, but this really shows us the potential and the power of this approach. So adoptive T cell therapy has been evaluated in clinical trials with real patients for a number of years now. There's two different types that I'll talk about, and the two that have really had the most experience so far. The first is called TIL therapy, and that uses tumor-infiltrating lymphocytes. It's a relatively simple procedure in that there's no genetic engineering involved. One just takes the T cells that are naturally found in the tumor, and I showed you examples of those earlier. You can just harvest those T cells, activate them in the lab, give them the right growth factors, and grow them up to very large numbers and give them back to the patient through an IV infusion. And that's just a way of really greatly increasing the numbers and activation state of the cells. And you can see that that achieves very impressive response rates in advanced melanoma. And there's also been some positive results reported in cervical cancer. Other cancers are currently under clinical trials, and the results so far are not as impressive in other cancers as originally seen in melanoma. So that's a real area of research to figure out how to raise those response rates. But nonetheless, that gives you an idea of one of very common therapy that's being tested today. And then the second example and I'll say more about it, I'll just briefly mention it now and give you details later, but is using something called CAR T cells, and in particular CAR T cells that have been engineered to recognize B cells, a marker called CD19, which have just had phenomenal success against certain types of leukemia and lymphoma, and have very recently received FDA approval in that setting. So we'll talk about CAR T cells in just a few minutes. In order to do T cell therapy, you need specialized infrastructure these days, and so at our center in British Columbia, we've just built a facility for clinical grade T cell production so that we can make these products and safely treat our patients. I'll just at this point mention, there is infrastructure required, and so this can be seen as a costly treatment. I think the way we do it today is fairly costly, but there are ways to bring those costs down in the future and to really simplify the manufacturing processes going forward. So it's a bit like the computers, when in the 1970s a computer filled up an entire room, and now the same capabilities are easily reduced down to an iPhone. I think similar gains can be made with T cell therapy. Once we figure out how to do it, we'll be able to simplify it. But this is kind of where we're at today. So just to give you an idea, this is a trial that we're planning at our center to treat relapsed gynecologic cancers. So that's ovarian, cervical, and endometrial cancer. Just to give you an idea how this fits into the rest of the patient's treatments, I'm showing you an example of an ovarian cancer patient. This is data from a real patient, although she didn't receive T cell therapy, but I'm going to show you how we could have tried to help her with T cell therapy. Instead, she received chemotherapy, as indicated here, which is standard of care. And you can see that that gave a good response. She actually went into remission. This is just a graph of her tumor burden over time. But she then had a relapse. As you can see, the tumor came back, had additional rounds of chemotherapy, which unfortunately she did achieve another remission, but you can see it was very short-lived. This is years after diagnosis, so just a few months. And then sadly the cancer came back a second time, and she was given further treatment, but that was not successful, and sadly she died of her disease. So this is the current kind of roller coaster that ovarian cancer patients go on today with our current treatments. And what our clinical trial will do, as indicated here, would be the patient would still have their primary chemotherapy, as shown here, because that is effective for most patients and they do achieve remission. In our trial, when the patient has relapses, at that point we would collect a tumor sample from the relapsed tumor and we would harvest the T cells from it, and we'd get busy growing those T cells in the lab while the patient went on and had their second-line chemotherapy, which we hope will help them, and we hope they never need their T cells, but unfortunately we know that most will. Most will, if they've relapsed once, they're at very high risk of relapsing again. But when that second relapse happens, we'd then be able to come in with our T cell therapy, which involves this. I won't get into the details, but there's a lymphodepletion step, then the T cells are infused and they get a growth factor called IL-2 to help support those T cells. And our goal would be to ward off, fight off that relapsed tumor. So this is what a clinical trial would look like. Eventually, once we get this really working, figure out how to use it, we'd of course like to move it more toward frontline. So perhaps trying to treat the very first recurrence, or possibly even combining it into the upfront care to prevent relapses from happening in the first place. So with clinical trials, we always start, for various reasons, we always start with sort of later stage disease, but once something starts to work, we move it earlier into the treatment course. What can we do to improve on this? It's in some ways you can think of it as a platform. Once you have T cell therapy going, you then have opportunities to come in and make it better. So there's ways that we can optimize the T cells by making them getting better at selecting out the tumor-reactive T cells very precisely. We can also genetically engineer the T cells to make them more potent, and I'll show you how we could do that. And you can think about optimizing the tumor as well, softening it up so that it's more receptive to a T cell attack. So there's drugs that can do that. And I'll show you briefly an example. We can use viruses, oncolytic viruses as they're called, is another way to, in that case, infect the tumor with the virus and cause an immune response that then these transferred T cells have better access to the tumor and a better opportunity to kill it. So all sorts of, I think, really exciting ideas for how to improve on TIL therapy. This is an example of how genetic engineering can help with this kind of approach. This is a study that we published a number of years ago, but really illustrates the point. So here I'm just showing a cartoon of all those signaling pathways we talked about earlier, the gas pedals and brakes that control a T cell. Well, there are internal brakes as well. So there's a protein called CBL-B, indicated here in red, which serves as a braking mechanism within the cell and basically puts the brake on a number of these signaling pathways. And CBL-B is there to protect us. It helps prevent autoimmunity, so that's important. But here's an opportunity where we can remove that CBL-B gene just in the T cells that are attacking the tumor. So when we do our adoptive T cell therapy and we have the T cells out of the patient's body in the lab, we can then engineer them to remove this CBL-B gene. And when that was done, this is in a mouse model of breast cancer, we did the adoptive T cell therapy with what we call wild-type T cells, or just the unmodified natural T cells. They don't do a very good job of getting into the tumor. And when we look at tumor growth over time, these tumors just keep getting bigger and bigger. The T cells had no beneficial effect whatsoever. But just by deleting or knocking out this CBL-B gene from the T cells, you can see the T cells now are much better able to get into the tumor. So we get very strong infiltration. And you can see the response here that these T cells eventually cause complete regression, complete disappearance of those tumors from the mice. So just by changing one part of the circuitry inside the T cell, we've gone from an ineffective immune response to a cure. So that's the potential of genetic engineering, and that's why we and others are excited about it. I'll talk now about chimeric antigen receptors, or CARs. I mentioned them earlier and said I'd give you more details. This is really a phenomenal area. It involves genetic engineering, and it's a great example of where we are today and what the future holds. So CARs, they're called chimeric because they have they're made up of two parts. They have an antibody-like region that is able to bind to or recognize the tumor, and then they have the signaling components of a T cell receptor, so a gas pedal on the inside. So these molecules are engineered into a T cell, so the T cell now on its surface has the antibody-like portion, and if that antibody recognizes a tumor cell, it basically is like putting the foot on the gas, and the T cell becomes activated and will attack the tumor cell. So these antibodies can be engineered to be against any protein, but where the biggest success so far has come is from a protein called CD19, which is expressed on certain leukemias. So we call these CD19 CARs. They redirect T cells to recognize these B-cell leukemias. And as I mentioned before, phenomenal successes, 90% complete response rates, with about 2/3 of those being durable responses in both kids and adults with leukemia, and leading to very recently to FDA approval for some indications. And then also very impressive responses in lymphoma, but also some room for improvement there, as we can see. So this is another revolution in cancer, the idea that we're now treating patients with genetically engineered T cells. Who would have guessed even 10 years ago that we'd get here so quickly? So in Canada, I'll just mention what we're doing in our research area. We're actually forming a national CAR T cell network funded through an organization called Biochemistry, where we're working, our groups in Victoria and Vancouver are working with a group in Ottawa to produce CAR T cells. So we're taking care of the viral manufacturing as well as the T cell manufacturing, and opening clinical trials up at both sites, and eventually inviting other cities in Canada to participate in this network. Not just Canada, but internationally as well. So we're expanding this to international collaborations. And the reason we're doing this is because we see so much opportunity here, and we think keeping this in the public sector or having an active public sector will be important to keep the costs under control, and also to allow as much innovation as possible. I'll talk next about how ideas for improving on CAR T cells. It's going to take a lot of work, and these CARs, we've got our current models, but it's going to be just like cars in the real world. Each year there's going to be new models coming out in the showroom with better and better features. And so we want to be set up to be able to contribute to that progress. So how can you make a better CAR? Well, 90% complete response rate is pretty good, isn't it? But there is room for improvement, and also we want to be able to build CARs that will attack other types of cancer besides leukemias and lymphomas. So what are some of the downsides with CARs, though? They're shown here at the bottom. A couple are related to side effects, something called cytokine release syndrome, which can cause serious problems for patients, although physicians are getting better at managing it. There are also neurotoxicities of unclear etiology or cause, which have led to some fatalities, obviously very serious. Patients also lose their healthy B cells. Those are important immune cells for as long as the CAR T cells are present. And as I indicated before, about a third of patients relapse. So they have a good response, but the tumor comes back, and often it has lost the target that the CARs recognize. We call these CD19 negative tumors. So these are some of the issues that need to be solved just in the setting of leukemias and lymphomas. Not shown here, though, is how do we also expand CAR T cells to treat other cancers like lung cancer, breast cancer, and so on. So one idea that we're developing in collaboration with Dr. Marty Boulanger at the University of Victoria is something we call mates, where we essentially take the CAR and we break it into two parts again. We separate the antibody component up top here from the receptor component shown down here, the signaling component, and then we stick them together again with, if you will, with a Velcro-like attachment so that they will come together and stick together, but they're not hardwired together. And what this somewhat simple change allows is that you can then give the patient the CAR T cells expressing just the receptor, but that receptor will not be activated unless the physician actually gives the patient antibody as an infusion. So the antibody portion becomes like a drug that you can give the patient when you want to activate the CAR T cells. But if there's any serious side effects, you can stop giving the antibody, and that will cool off the whole reaction. Or if the patient is cured of their leukemia, let's say, and you don't need the CAR T cells anymore, just stop giving the antibody altogether, and those CAR T cells will no longer be activated. So it's a simple idea, but it then gives a very much better control over the whole reaction. So I just put that up there as one idea for how to make CAR T cells better. So just I could give you just one other very brief example. I'm not going to go through all of these, but just I wanted to give a concept of the diversity of ideas out there. And so I'll just end by talking about oncolytic viruses, which is, I think, a real up-and-coming part of the immunotherapy landscape. It has also achieved some success recently. So oncolytic viruses are viruses that are either selected or engineered to recognize and attack tumors. So oncolytic meaning cancer busting. So this top panel shows what happens when a healthy cell is infected by a virus. You see the green virus going in there, but in a healthy cell, these oncolytic viruses are such that they're not successful inside a healthy cell. They can infect them perhaps, but they're not able to replicate inside them due to differences in their signaling properties. But the viruses are selected such that when they find themselves inside a cancer cell, shown here as the purple cell, that's just the perfect environment for them to replicate. They've got all the right signals and other factors that they need, and so they replicate like crazy and eventually kill that cancer cell, and releasing all those viral particles, which can then go and do the same thing to the next cancer cell along. So that's the basic idea behind oncolytic viruses and what everybody hopes that they will do. But what's been learned from working with them is that they do something else in addition, which is that they stimulate an immune response against the tumor. The immune system looks at this and says, wait, there's some sort of viral infection going on over here, and the T cells and B cells come rushing in to fight the virus, and in so doing, they then notice the tumor and start attacking it as well. And so one type of oncolytic virus is known as TVEC. I won't try and say the full name here, indicated up here. This was just received FDA approval for the treatment of melanoma. And with TVEC, what was done is they wanted to really enhance this immune stimulatory effect. So they introduced a gene for a molecule called GM-CSF, which we won't get into the details here, but it actually helps really promote a stronger T cell response. And so you get not only the direct killing of tumors by the virus, but you stimulate the immune system. And that's important because what it means is that you can perhaps inject the virus into one tumor in the patient, but by triggering an immune response, those T cells can then go and kill off tumor deposits elsewhere in the body that the virus might not be able to get to on its own. So you go from a local effect to a systemic effect. So here's a picture of what these oncolytic viruses can do. This is work from my lab, just to make the simple point of how they can trigger an immune response. So this is an untreated tumor on the left. Again, there's no T cells in these tumors at baseline in this particular model. Then after infecting this tumor with an oncolytic virus called VSV, looking a few days later, you can see that the T cells have come rushing in, all those brown cells, and they are fighting the virus, no doubt, but also attacking the tumor. And in our mouse models, we can actually get complete regression of these tumors by infecting them with these oncolytic viruses. So in closing, the current treatments for cancer that are familiar to everyone: surgery, radiation therapy, chemotherapy, hormone therapy. Some of the newer drugs are often referred to as targeted therapies. But what we have here now, adding to this, is immunotherapy. This slide used to say coming soon. It now says new, because it's actually here, as I've shown, and with all these FDA approvals, lots of potential for the future as well. That is replacing chemotherapy in some settings, might replace some of these other therapies, but it's not a competition here. It's really about finding the right combination for each type of cancer. I think all of these other treatments will still have their place in the future, but immunotherapy will certainly be a big part of things. So it's really an unprecedented opportunity. We're seeing long-lasting curative treatments, not curing everybody, far from it, but some really good signals, lower side effects overall, wide applicability, natural mechanism of action. So we're working with the body's own immune system. And it also leads to opportunities for locally produced T cell therapies, as we talked about, which creates jobs, talent, innovation, intellectual property, can help with cost control. So from the socioeconomic side of things as well, immunotherapy, especially these adoptive T cell therapy, offers these additional benefits. So I'll just show, these are the many people in my lab and various collaborators who I've worked with over the years. Haven't covered everybody's work here, far from it, but just want to acknowledge a large team that I'm fortunate to work with, both scientists and clinicians across Canada and around the world. And we have our various funding organizations as well. But most of all, I want to thank our patients who contributed so generously tumor samples and blood samples for our studies, and all the patients around the world who are participating in these clinical trials of immunotherapy and have allowed these breakthroughs to really happen and show the world the potential of this new approach to cancer treatment. And I'll close there and open this up for discussion. Thanks very much for your attention.
Thank you, Dr. Nelson, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your answer into the box that appears on your screen and click on the send button. We'll answer as many questions as we have time for. Okay, Dr. Nelson, our first question of the day asks, what kind of side effects are associated with immunotherapy?
That's an excellent question. Yeah, I hinted that there were some, and with CAR T cells, we talked about some of the serious ones, including this neurotoxicity leading to death. That's as serious as it gets, and are a matter of concern. Let me back up, though, and just talk about, let's start with these checkpoint therapies. The first thing I talked about is drugs that affect the brakes on the immune system. Those generally have, for some patients, there's no side effects, and for others, there's sort of inflammation that's caused. Autoimmune-like syndromes can be caused. But all of those are transient in the vast majority of patients. And so a patient might experience these while they're on treatment, but they resolve with time. So fortunately, we're not seeing immunotherapies causing long-term autoimmunity in patients. But nonetheless, while on treatment, some patients might experience things like colitis, so inflammation of the bowel, for example. And not to minimize that experience for the patient, but it is temporary, and they can move on and more or less get back to normal after that. With the CAR T cells, the toxicities can be more significant, but clinicians are learning quickly how to manage these and prevent bad outcomes. And I gave examples of how we think with better engineering we can gain better control of these reactions and minimize things much further. So overall, the toxicities are manageable and certainly no worse than we see with other cancer treatments.
Okay, thank you for that information. The second question today asks, what are the costs associated with immunotherapy?
Yeah, so I touched on that, but I'm thankful for the question just to say a bit more about it. In general, these checkpoint antibodies or ballpark figures are about $100,000 per patient. That's not out of line with other modern cancer treatments, so it's not, might sound like a lot to some people, but it's not considered crazy money in oncology. But sustainability for our healthcare systems is a big issue. The more successful these agents are, the more patients will receive them, and that creates costs for the healthcare system. And whether it's a private or public health care system, somebody has to figure out how to pay those bills. So if we're talking about combining these different immunotherapies, those costs could really stack up. And I think it's fair to say every healthcare system is concerned about this and how to pay for it. My view is, like anything, things usually start out expensive and then they get less expensive with time as we figure out how to use them better, how to innovate further. Things come off patent eventually. That will bring down costs, allow market for generics. And I think with T-cell engineering, I've tried to make the case we could, by just changing the circuitry of the T-cells, you might be able to eliminate or reduce the need for some of these other expensive drugs to give at the same time.
Thank you for your answer. Let's see. The third question of the day asks, will immunotherapy eventually work for all types of cancer?
Great question. So I showed quite a diversity of cancers that are responding to checkpoint blockade, for example. The cancers that are responding today to today's drugs, in general, one thing they all have in common is they have a very high mutation load, as we say. By that I mean they have a lot of mutations in them. At the very beginning, we talked about how cancers acquire mutations, and I showed you some of those chromosome pictures and how messed up the cancer genome becomes. Somewhat ironically, the more messed up the cancer is, the better it responds to immunotherapy. And the reason for that, we think, is because the more messed up a cancer is, the more foreign it looks to the immune system, and the more willing the immune system is to attack it, the more able it is to attack it. So other cancers, such as ovarian cancer, which I talked a lot about, in general don't have that many mutations overall, and so they're not responding as well to today's immunotherapies. So, but what about the future? I think there's lots of potential using, for example, CAR T cells to, if the tumor doesn't look messed up enough, we can still teach the T cells or engineer them to recognize other features on the tumor other than mutations that would allow for a good immune response. So in theory, there's no reason to think immunotherapy couldn't be beneficial for every type of cancer, but time will tell which cancers prove to be the most sensitive.
Thank you. Looks like we have time for one more question today, Dr. Nelson. And this question asks, what can I do to strengthen my immune system?
This is a question I get a lot from patients. When they hear my presentation, they say, well, also some of them say, I had cancer 20 years ago and I'm still alive, and I've taken this particular extract all these years, or I've done yoga or this or that, and I'm pretty sure that's why I beat my cancer. And to people who have survived and have those beliefs, I say, good on you. It's not for me to say otherwise, because I think the patients know inside themselves what's working for them. So I'm just a scientist that tries to make things better, but if you're alive, then there's nothing, like, you can't do much better than that. So patients know to some extent, but then some also want to know, is there practical advice I could give? And in general, there's not a lot of scientific evidence for all the immune-stimulating nutraceuticals and things that are out there, the various products you'd find in a health food store. Many of them claim to boost the immune response. For very few of them, if any, have really hard scientific evidence for that. Now, again, that doesn't mean they don't work or don't help some people. I don't mean to be dismissive of it, but I'm a scientist, and scientists, we like to answer questions by doing experiments, and let's give 1,000 people this agent and 1,000 people a placebo and see what happens. So for most of these nutraceuticals, those kind of really rigorous scientific studies haven't been done. So if you're wanting to have a science-based approach to life, those wouldn't offer solutions. On the other hand, again, if anyone's taking them and thinks they're benefiting from them, then don't let me tell you otherwise. But what do I do in my own life? I think your immune system, we all have a bit of a barometer for it, which is we all know when we're feeling tired and run down and we're more likely to pick up that cold virus or something. We all go through those phases that we're feeling stressed. That's your barometer telling you your immune system probably is on a low gear and not firing optimally. So what do you do to get out of those kind of states? Get a good night's sleep, eat healthy food, get some exercise, lower your stress, get back to feeling really good and strong, just as as a person, and that's when your immune system is really doing the best. So in order to take it to a supercharged level, I think it's hard to do that with lifestyle and everything I talked about today with immunotherapy. I think that's where science can really come in and help supercharge things. But otherwise, it's really just about following your good advice about healthy living.
Well, thank you so much, Dr. Nelson, for your presentation. And I would also like to thank you for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again here soon at LabRoots. Goodbye.